Welcome to another biology video. So in our today's discussion, we're going to discuss molecular biology and genetics question. This was an exam which was written in 2021. So we have question 1. So question 1 is saying use the following information to answer questions 1 to 5. So from the look of things, this question is coming from genetics. And it's a chi-square. Okay, it's a chi-square. So for us to answer these 5 questions, we have to conduct what is known as a chi-square. So let's first briefly understand what the chi-square is, because we cannot answer this question minus understanding or knowing what the chi-square is. So we'll come back to this question. Let's do this. So what is a chi-square or chi-square test? So a chi-square test, this is a statistical test which is used to confirm whether the differences between the observed and expected values are significant or not. It is a statistical test which is used to confirm whether the differences between the observed and the expected are significant or not. So we are going to have what is known as observed values. You're always going to have observed values. Then you have to come up with the expected values. So these expected values, they are derived from Mendelian ratios. Your understanding on Mendelian ratios is what is going to help you come up with the expected values. So these observed values, these are going to be the values which are going to be given in the question or in the statement. The numbers or the figure that you are going to see in the statement, those are your observed values. Then based on the type of cross which was conducted, you come up with the ratios. The ratios that you are going to find, they are the ones now which are going to help you to come up with the expected values. Upon you finding these expected values, you are then going to calculate for the chi-square. The symbol for chi-square is x squared. So when I write x squared, I simply mean chi-square. So you calculate for the chi-square value. So now, when you calculate for the chi-square value, you are going to compare this chi-square value to another value which is known as the p-value. So the p-value, or before I come to the p-value, let's first do this. So if you have a cross, let's say this was being close to that, you know that this is going to be, let's say this is tall, was close to dwarf or short. Okay, so if you conduct this particular clause, you know that from this clause the expected ratios is 1 to 1. That is, it's 50-50 kind of thing. So if from this clause 50 plants were produced, you are expecting that out of 50 you're expecting 1/2. So I'm using these ratios. So I'm using these ratios to come up with the expected values from that clause. I want you to understand how you calculate for the expected values. So you're going to say 1 out of the total 2. So you add these. For you to calculate for the ratio, that's what you do. So 1 out of 2 multiplied by the total 50. So you're going to get a 25. The same with this one. Those which are expected to be short, it's 1 out of 2 as well, multiplied by the total 50. So you are also going to get a 25. It means that in this case, out of 50, you are expecting 25 to be tall and 25 to be short from this kind of a cross, and this is a monohybrid cross. So now, you're going to be given a question. Then the statement, let's say the statement is telling you that from a cross, 50 plants were produced, and out of 50, it was observed that 30 were tall and 20 were short. 20 were short. So test whether the difference between the observed and the expected is significant or not. So that's where now you're going to conduct a chi-square. So these numbers are the observed values. Then you have to come up with the expected values, of which you have already calculated. They are here. So if you look at these numerical differences, the expected is 25, observed is 30. The numerical difference there is 5. The expected is 25. The observed is 20. The numerical difference there is also 5. So you have to test whether this difference between the observed and the expected is significant or not. This is where you have to calculate for the chi-square value. Okay? This is the first step we should take. So, after you calculate for the chi-square value, of which we're going to go step by step on how we calculate for the chi-square value. So here I'm just giving you an overview of all questions involving chi-square, what you are supposed to do. So you're going to calculate for the chi-square value. So this chi-square value, when you calculate it, you are now going to compare it with what we call the p-value. So the p-value, you obtain it from the statistical table. The p-value is also known as tabulated value or critical value. All these mean one and the same thing. So this value, you obtain it from the statistical table. The statistical or the chi-square table is always given where this question is concerned. As long as there's a question on the chi-square, they are always going to give you a statistical table. So from that statistical table, that's where you are going to obtain this value. Okay? You calculate for the chi-square, then you are supposed also to have the p-value. So this p-value is the one which you obtain it from the table. Now, how do you pick this value? This value is always tested at a probability of 5%. So we're testing the difference between the observed and the expected at 5%. 5% is the same as 0.05. So this is the probability at which you pick this value. So we have probability this side. Then it's also picked at a degree of freedom. So there's what we call degree of freedom. So this degree of freedom is given by degree of freedom as a formula, which is equal to n minus 1. So what is this n? So n is the total possible traits or phenotypes. Total possible trait or phenotypes, that's the n. So from the cross that we had earlier, we had this cross. So from this type of a cross, how many different phenotypes or traits are you expecting to get? This is a monohybrid cross. So we're expecting to get only two traits or phenotypes. That is tall and short or dwarf. So these are going to be the total possible number of traits or total possible number of phenotypes. So it means that if you want to calculate for the degree of freedom in this case, your degree of freedom is going to be 2. So it's going to be, or your n is going to be 2. 2 minus 1. So your degree of freedom is going to be 1. So now you want to obtain this p-value from the statistical table. So you're going to have degrees of freedom, this going down. Then you're going to have probability this side. So now you're going to have these 1, 2, 3, and so on. So in our case, our degree of freedom is 1. So where this 1 and this 0.05 correspond, that is going to be your figure. That's the figure you pick. So let's say it's here. So if this figure, let's say it's 3.4, you are just going to pick this value. You don't need to stress. Just check your degree of freedom and the probability, which is 0.05, where the two intersect. That figure, that's your degree of freedom. So you just pick it. It means that in this case, our p-value is going to be equal to 3.4. So now you have to compare your calculated value, which is the chi-square, and this value for you to make a difference, or for you to make a conclusion based on the question being asked. So I've given you an example of dihybrid cross, how you obtain the degree of freedom. What if you have that was monohybrid cross. What if now you have dihybrid cross? So you know that most of the times in dihybrid cross, the expected phenotypes are going to be 4. it means that your degree of freedom is going to be equal to n minus 1. So it's going to be n is 4 minus 1. This is going to be equal to 3. It means that your degree of freedom, most of the times when you're dealing with dihybrid cross, your degree of freedom is going to be 3. Then 3 as your degree of freedom at 0.05, then you pick your figure. That's your p-value. So now, how do you make a conclusion based on the figures which you are going to find? So you are going to calculate for this. So let's say we have the p-value of 3.4. We have obtained it from the statistical table, and we have calculated for the chi-square. So we find that the chi-square value is 3. The chi-square value, let's say, is 2.3. So what conclusion are you going to make, or what does this mean in genetics? So there are two possibilities, or let me say 3. The first one is if the chi-square is greater than the p-value. If the chi-square is greater than the p-value, let's say if this chi-square was 5.8, then you find the p-value to be 3.4, just like this. So the chi-square is 5.8 and the p-value is 3.4. What conclusion are you going to make, or what does that mean? So we have this situation in that case. So if the chi-square is greater than the p-value, in this case what it means is that there is a significant difference between the observed and expected values. Remember that we said the chi-square test is a statistical test which is used to test whether the differences between observed and expected values are significant or not. So in which case are you going to say that the difference between the observed and expected is significant or it's not significant? So these are the scenarios I'm giving you now, or how you are going to conclude your results. So if the chi-square value is greater than the p-value, it means that there is a significant difference. There is a significant difference between the observed. There's a significant difference between the observed and expected values. And in that case, the hypothesis is going to be rejected, or the results are going to be rejected. In other words, there will be rejection when the chi-square value surpasses the tabulated value. Another scenario is if the chi-square value is now less than the p-value, like in this case 2.3 as your calculated value is less than 3.4 as your p-value. So what conclusion are you going to make? In this case, when the chi-square value is less than the p-value, it means that the difference between the observed and expected values is not significant. It means that these values are within the expected Mendelian ratios. When the chi-square value is less than the p-value, it means that the values are within the expected Mendelian ratios. So in that case, where we had expected 25, and observed we had 30 to 20. So if you conduct the chi-square and you find that the chi-square is less than the p-value, regardless of these numerical differences, you are going to say the values are within the expected Mendelian ratios because the chi-square is less than the p-value, and the results are going to be accepted. The hypothesis will be accepted, in other words. So these are the conclusions that you make. Then if they are equal, although I've never come across a situation where chi-square equals the p-value. So when they are equal in this case, there's going to be rejection. There's going to be rejection. The results will be rejected. It also means that there's a significant difference between the observed values and expected values. So when they are equal, it's the same as chi-square is greater than the p-value. That is the conclusion which you are going to make. So overall, with respect to the chi-square or problems relating to chi-square, these are what you are supposed to know as a foundation or the basics. So now we can go ahead and answer this question. So when answering the chi-square, there are certain things which you have to bear with in mind, or you have to know. You know to say that because these calculations are based on the findings of Geiger-Mendel, so they are standard. They are those standard ratios. You know that when Geiger-Mendel closed tall plant with dwarf plant, all the F1 generation were tall, right? So all of them were heterozygous. 4 of them were this, like this. So there are two things here now. One of them is that he conducted a test cross. A test cross that is, so in the F1, first of all, this phenotype, which is dwarf, disappears. It is on this one, which is dominant, that takes over the F1. So because of this, Geiger-Mendel wondered, like, what would have happened to this short plant? So for him to know the genotype or the F1, he now did what is known as a test cross. A test cross, this is a type of cross where you are crossing one of the parents from the F1 to or one of the offsprings from the F1 to one of the parents which has got a recessive trait. In this case, it's this one. So test cross here, you're going to pick one of the F1, that is going to be this, cross it with the parent with a recessive trait, like this. When you conduct this cross, you're going to have, when you conduct this cross, I think what are going to have 1-to-1. Yeah. So when you conduct this cross, we're going to have the ratios which will be 1-to-1. So a test cross in monohybrid cross, the Mendelian ratios are always 1-to-1. Then the other thing what he did is he allowed the F1 to cross amongst themselves. So when you allow the F1 to cross amongst themselves, it means there now you are crossing this, cross it with this. The ratios in this case are going to be 3-to-1. These are going to be 3-to-1. You can conduct a cross, but this is what you are going to get. 3 are going to be tall and 1 is going to be short. So this is when you allow the F1 to cross amongst themselves. So this is going to be the F2. The ratios are 3-to-1. So these are the ratios which we call the expected Mendelian ratios. These are the ratios which we are going to use to calculate for the expected values. Okay, if it's in monohybrid course. The same was true when he conducted a dihybrid cross. We know to say that Geiger-Mendel crossed garden peas. So he crossed round yellow garden peas. These ones were crossed to wrinkled green garden peas. So what he observed is that all the F1 in this dihybrid cross, all the F1 were round and yellow. All of them had this genotype. So in this case, because it's dihybrid cross, you're going to have 16 offsprings or progenies. And all of them are going to be round and yellow. So this is going to disappear, which is wrinkled and green. It will disappear in the F1. So again, he was not certain of the genotype or the F1. What did he do? He crossed one of the F1 progenies to the parent with a recessive trait. So he did this, crossing one of the progenies from the F1 with a parent of a recessive trait. So this is a test cross in dihybrid cross. This is a test cross. So you are crossing to the parent with a recessive trait and one of the F1 progenies. That's a test cross. When you cross this one to the parent with a dominant trait like this one, that is a back cross. It's not a test cross. So a test cross is also a back cross, but a back cross is not a test cross. Back cross is crossing to any of the parents with one of the progeny from the F1. But test cross is restricted to the parent with a recessive trait. That's a test cross. So after crossing these, what we obtained were that all the ratios were 1-to-1 to 1-to-1 because they were 4, 4, 4. You know that from dihybrid you are going to have 16. So when you reduce these, you are going to have these ratios. So these ratios are what we call expected Mendelian ratios, and you should know the type of ratios which you are going to get in dihybrid test cross and monohybrid test cross. Then again, another scenario that you are going to allow the F1 to cross amongst themselves. So it means that since all the F1 are going to have the same genotype, you are now going to allow them to cross amongst themselves. So when you allowed this to happen, what he obtained of course 16 progenes, but these 16 progenes were in the ratio 9 to 3 to 1. So these are also expected Mendelian ratios when you allow the F1 to cross amongst themselves. The expected Mendelian ratios are these, and these are the ratios which you are going to use to calculate for the expected values. So then later on you are going to make conclusion if there is a significant difference between the observed and the expected values. So that's what we are going to have. So now we can go ahead and answer this question. So we have it here. So it's saying use the following information to answer questions 1 to 5. So the question here is a male fruit fly, which is Drosophila melanogaster, with black body and long wings. So as you have seen, this is the genotype that we're going to use. So we have that as the genotype was mated with a female with a white body and a vestigial wing. So this is the genotype for that. Now from here you can see that black is dominant over white, because white is recessive, can only be expressed if we have homozygous recessive alleles like that expressed together. Then also long wings is dominant over vestigial wings, so it can also only be expressed if these recessive alleles are expressed in a homozygous form. So that's what we have. So from here, if you are to conduct a cross between these two, one thing you are going to observe is that if you are to conduct a cross from here, if we are to cross these to get the F1, what you are going to observe is that all the F1 progenes are going to have this genotype. All of them are going to have this genotype. That will be they are going to be 16 because this is a dihybrid cross. So all the F1 are going to be, in terms of phenotype, they are going to be black. They are going to have black body with long wings. This is going to disappear. The white body and vestigial wings is going to disappear. This is similar to what, or it's just the same to what Gregor Mendel did when he crossed round and yellow garden peas with wrinkled and green garden peas. All the F1 were round and yellow. So this is what we are getting from here. It's very similar. So now the question goes on to say the F1, that is the progenes which we have obtained. Since we are saying all of the 16 are going to have this genotype, so the F1 flies with this genotype, as you have seen, we are mated with white body vestigial winged flies. So this is the genotype. Where do we see this? It's the original parent to the progenes. So it's this one here. So the cross now is between one of the F1 progenes. We obtain one of the F1 and cross it to the parent with a recessive genotype. That one. So what is this? This is what we call a test cross. So that's a test cross already. And we know the expected ratios, the expected Mendelian ratios when we're conducting a test cross in dihybrid cross. Then they are saying their offspring, the F2 generation. When you cross this one now, you are going to obtain the F2 generation, appear as indicated below. So these are the results which were obtained after conducting this cross. So these numbers which you see here, earlier on I mentioned something like you are going to have observed and expected. So I said the numbers that are going to be given in the statement or the question, those are what we call observed values. So already here our observed values are the ones we have here. Then we should come up with the expected values. Where are we going to obtain these expected values? These are going to be obtained by using the ratios which we are supposed to find. Since I already know what the ratios are going to be here, I would have gone straight forward to calculating for the chi-square. But we are going to go step by step on how you come up with the expected values. Then later on we are going to calculate. So from here what you do is you conduct a Punnett square. I already know what we're going to obtain after the Punnett square. But we just have to go step by step so that you understand. Because not all the questions are going to be based on the test clause. No, some they are going to tell you to say the F1 we are allowed to test clause amongst themselves. So the approach may be different and the ratios may differ. So what you do after this, since you already have the observed values, you now have to calculate for the expected values. How do you calculate for the expected values? You have to know the ratios. And you can only know the ratios if you conduct the Punnett square, because this is a dihybrid clause. So what do we have? So we have this genotype, that is black and long wings being closed to white body and vestige wings. So when you want to conduct the Punnett square, you are supposed to come up with the gametes, two sets of gametes, 4 from each set. So these are going to give us 4 gametes. These will also give us 4 gametes. So we're going to have 2 sets of gametes, 4 from each. So how do you come up with the gametes? This is going to go with that one. The first gamete will be this. The other one, this one should also go with that one. So we are going to obtain this. You can't close this one with this one because they are alleles of the same gene. So you only close alleles of different genes. Then you start with this one again. This one should go with that one. So this is going to be another set of genes. Then this one should go with that one. That will be another set of genes, like this. The same this side. So this side we're going to have 4, which will be the same, because now you know how we are coming up with this. Okay? So this one should go with this one and that one. You're going to have 2 of those. The same this one and that one, so you obtain these. So now we have 2 sets of gametes, this set and this set. So these sets of gametes which we have now, you are going to say one set is male, the other set is female. It doesn't matter which ones you choose to be your female gametes and which one you choose to be your male gametes. So in my case I'll choose this. It doesn't matter though. Then now you're going to feed these in the Punnett square. Okay? We're going to feed these in the Punnett square so that we can come up with the progenes. After we find the progenes, then we have to calculate we have to come up with the ratios. So now we are going to say we have this and that. Then you feed in the gametes, one in each. So I'll start with this set of gametes like this. Since there are 4, we are done. That other set of gametes, you feed them in here. So there we had this, then we had this, and also this and this. Oh, sorry. So this one will be this. So then when you are going to have to obtain the offsprings, you feed them in there. Okay? So we start. The first one is going to be this and that. So we are going to have this, that, and this. So as you have seen here, we have got BL here. So when coming up with a progeny, you can't have something like this. No, you can't have something like this. So what you do is alleles of the same gene, they should be together. So these are alleles of the same gene, which codes for body color, encodes for body color. Then these encodes for body size or shape. Okay, so this is how you are supposed to. This is what you are supposed to do, and not this. Then you continue. So let's continue so that we fill up this. So we are going to have 16 of them. This is also going to be like this. Then the same here, Then last 3 the same. I'm done with that one. I'll come here and we'll have this the same, even here, even here. So it's not always that they are going to come out like this. It depends on the genotypes that you have been given. So you just multiply as long as your gametes are correct. These are going to be simple things to do, and you now know how to come up with the gametes. Then the last ones. Then finally, so we have filled this Punnett square. So if you count these, they are supposed to be 16. Now all the 16 here, we are expecting to have four different phenotypes. So the first phenotype is all those which have got this genotype. All those which have got this genotype, these are going to be black with long wings. So say black, long. Then those with this genotype, these are going to be black and vestigial wings. Then those with this genotype, these are going to be white with long wings. And the last 3, we are going to have those with this genotype. As you have seen, we have all these genotypes. So these are going to be white with vestigial wings. Now come up with the ratios. Out of 16, how many of those have got this kind of genotype? So you go back and start counting. So we have 1, 2, 3, 4. We only have 4. We don't have any other which has got at least dominant B and dominant L. We don't have. They are only 4. So you're going to say these are 4. You come to those which have got this genotype. Those which have got this genotype, they are 1, 2, 3, 4 as well. They are only 4. You can check out, you just find that they are just 4. Then those which have got this genotype, so we have 1, 2, 3, 4 as well. Those which are going to be white with long wings, they are also 4. So we are going to say this is 4. Then last 3, those with this genotype. So as you can see, we have 1, 2, 3, 4. They are also 4. So we're going to say this is also 4. So now these ones, if you add, they should give you 16. So they are in the ratios 4 to 4 to 4 to 4. These are the phenotypes. If you reduce these, since 4 can go into all these numbers, you are going to have 1 to 1 to 1. So these, since I told you to say this is a test cross, and the expected Mendelian ratios in the test cross is always 1 to 1 to 1 to 1, like that. So now we have the ratios. Since we have the ratios, we can now calculate for the expected values. So how do you calculate for the expected values? For the expected values, we are going to say, let's say those ones which are expected to be black with long wings, it's 1 out of the total 1, 2, 3, 4. So the ratio is going to be 1 over 4 multiplied by, then you have now to add the total number of flies which were produced from this cross. So what are we going to do? We go back, we come back here. Let's add the observed values. These observed values, they are going to give us the total number of progenes which were produced in that cross. Okay? Which were produced in that cross. So what do we have? So we have so what do we have? So we are going to have we are going to add, okay, so just a minute. Okay. Okay, so okay, so we're going to have okay. All right, so there was a technical error, but we continue. So I was saying that for us to calculate for the expected values, we should first add this one so that we get to have the total number of proteins which were produced in this cross. So we're going to add the 210 plus 206 plus 50, then plus 46 plus 46. So the total is 512. So the total we have 512. Now from 512 we can calculate for the expected values. So since the ratios are 1 to 1 to 1, what are we expecting from this? Just divide this because it's going to be 1 out of 4. For each it's going to be 1 out of 4 multiplied by 512. So if we say 1 out of 4 multiplied by 512, we're getting 128. So it's going to be 128 to 128 to 128 to 128. All of them are going to be like this because they are in the ratios which are the same. So now these are the expected values. We have obtained the expected values. At this point now we have both the expected values as well as the observed values. Now if you are to check here, you can surely see that here the observed are 210, but the expected are only 128. The expected for those which are supposed to be white and vestigiling, they are 206, or the observed, but the expected are only 128. These are 50, expected is 128. These are 46, expected is 128 as well. So there's a numerical difference, but for us to tell if the difference between the observed and the expected values is significant or not, we have to conduct the chi-square. So we now have this table has helped us to come up with the ratios. That's why you are supposed to conduct the Punnett square. The Punnett square is going to help you to come up with the ratios. Once you come up with the ratios, you are good to come up with the expected values because you are going to use these ratios. 1 out of 4 multiplied by 512, you are getting 128. Now, in a case where the ratios, let's say if these ratios were 9 to 3 to 3 to 1, you are not going to say all of them are the same, no. So, in that case, we would have said 9 out of 16 multiplied by 512 for these ones. Then, for these ones, you would have said 3 out of 16. 16, you are adding all these numbers. So, you're going to get 16 multiplied by the total 512. Then this one will also be 3 out of 16 multiplied by 512. This one was going to be 1 over 4 multiplied by 512. That's what you do. But in our case, we have the ratios which are 1 to 1 to 1 to 1. That's why the numbers are the same throughout. If the ratios were like this, the numbers were not going to be the same. So now we have the expected values as well as the observed values. So we can calculate for the chi-square. So the chi-square, you need another table. So this table for the chi-square will look like this. Okay? It's similar to the one for chi-square, but what you feed in here is what is different. So here you're going to have a trait. So trait, I said this is a phenotype. Then after the trait, you're going to feed in the observed. So observed values. After observed, you're going to have expected values. Then the difference, observed minus expected. Then next you square, observed minus expected squared. Then here, this is what is going to give you the chi-square. Observed minus expected squared. divide by expected. So you now see the reason why we had to calculate for the expected, because we need them here as well as there to calculate for the chi-square. This is what we need. So at the end, for us to calculate for the chi-square, we're going to add what we're going to have here. Adding, we're going to get a number here that is going to be our chi-square. So let's start. So we had black, some black with long wings, black with long wings. Then we're also going to have black with V-stig wings. Then we are now going to have white with long wings. Then finally we are going to have white with V-stig wings. Then go back and obtain the observed values. Just copy them the way they are. How many were black and long wings? So we go back. How many were black with long wings? So the first one. So that is 210. So we have 210. Those which were black with long wings were 210. What about black with V-stig wing? 46. So black with V-stig wing only 46. So we go back, 210 black long, and so these were 210. Then black with V-stig only 46. Then we go back also white and long and white and V-stig. So white and long, those which were white and long. So we have white and long 50, while white and V-stig 206. 206. So white V-stig 206, the other one 50. White V-stig 206, so I'll say 206 here. Then these ones were on 50. So the observed, we just feed them the way they are. And this order here doesn't matter. What matters is these numbers. You have to be very critical. If you wanted, you would have just followed, but copied the way they were on that side. But I couldn't see. So we have the observed values. Then what are the expected? The expected, we have already calculated that the ratios were 1 to 1 to 1 to 1. So each one of these is 128. And when you add these, they should give you the total, which is 512. 128. 128 for each. So you need the calculator here. Then now we are going to say observed minus expected. So for the first one, observed we have 210 minus expected is 128. So we have 82. Okay? 82. Next we have to square this difference. So when we square this, we are getting 6,724. Next we are going to say this is squared divided by expected, which is 128. So what are we getting? Divided by 128. So we are getting 52.53. So we are getting 52.53. Then we come back this other one. 46 minus 128. 46 minus 128 is negative 82. This negative 82 squared, we are getting the same 6,724. Divide this one by expected, which is 128. So divide by 128. So we're going to get the same, which is 52.53. We have 52.53. When going to write an exam or a test where these are involved, just go there with a calculator, because the chi-square will always demand you to use the calculator. Then 50 minus 128, we are getting negative 75. Negative 75 squared, this is giving us 5,625. Divide this one by expected, which is 128. So I'm getting 43.9. I'm getting 43.95. Let me repeat this. 50 minus 128. So we are getting 78. Yeah, it's 78. $50. So we have 50 minus 128. I'm getting negative 78, not what I wrote earlier. Negative 78. We square this, I'm getting 6,084. 6084 divided by expected 128, I'm getting 47.5. 47.53. Then the last one we have 206 minus expected 128, that is 78. I square this, we're going to get the same 6084. Then divide this one by the expected, which is 128. So I'm getting 47.53. So now we have calculated. For us to come up with the chi-square, just add these numbers. The value that you are going to get, that is your chi-square. So we're going to say our chi-square value will be equal to, so let's add. So we have 52.53 plus 52.53 again, plus 47.53 plus another 47.53 total. So I'm getting 200. So I'm getting 200.12. So now we are done. We have calculated for the chi-square. So our chi-square value is equal to 200. Okay, this is the same as 200 point. Okay, we can leave it as 200. Okay, when we round it off, the point will be ignored because this is not the point are not significant than less than 4, or they are less than 5. So our chi-square is 200. Then now let's obtain the p-value. Since we say the p-value is obtained from the tabulated table, or from the statistical table, what is going to be the p-value? So we said p-value is obtained from the statistical table. So let's, in this case, the statistical table which was given it, where is it? It's here. So here it's saying a chi-square test was then carried out. This is a continuation of the question. Was carried out to test the results of the progeny using table 1. So table 1 is the one we have here. So table 1 is saying tabulated or critical chi-squared values for up to 4 degrees of freedom. So as you have seen, this table has got these degrees of freedom, this side. Then it has got a probability. So probability at different probabilities. This is a 1%, 5%, 10%, 20%, just like that. But our interest, we say that it's 5%, which is the same as 0.05. So our probability, you obtain the p-value at the probability of 0.05 or 5% and at a degree of freedom. So we said that degree of freedom is given by n minus 1. What is our n in our case? So we said n is the total possible number of traits. So we have 4 different phenotypes in that cross. So 4 minus 1. It means that our degree of freedom in this case is going to be equal to 3. So where is 3? So 3 is here as our degree of freedom. And also the 0.05 is there. So where the 2 intersect, that is our p-value. So the 2 intersect in here. So what is our p-value? Our p-value is 7.8. So we have the p-value of 7.8. So now we have both. This is what we have been looking for. All this process was just for us to calculate for this. For us to arrive at this point, you are supposed to undergo all these processes. You can't skip. Then the p-value, this one you can even find it even before calculating for the chi-square. Because it's just the n minus 1, the degree of freedom. If you know the n, then you are going to come up with the degree of freedom, and you know it's at 5%. So this one you can always find. And this is, I always call this as a free mark in an exam because there's no need of you thinking or doing anything but the chi-square. So in this case, what conclusion are you going to make? Is there a significant difference between the observed and expected values? So which scenario amongst the 3 that I gave you do we have here? So what we say that 200 is greater than 7.8. So the chi-square is greater than p-value. So in this case, the hypothesis is going to be rejected. The results will be rejected. Why? Because the chi-square is greater than the p-value. What does it also mean? What it means is that the ratios are not within the expected Mendelian ratios, or the values are not within the expected Mendelian ratios, because the chi-square has surpassed the p-value. What does it also mean? It also means that the difference between the observed and expected is significant. When the chi-square is greater than the p-value, it means that there's a significant difference between the observed and the expected values. So these are some of the conclusions that we can make from this kind of calculation. So we can now go back and continue answering these questions because we have everything. Okay. So the table which we had, the question is this, the table is this one. So sometimes you're going to have this question, but this table will not be where the question is. So this table is always attached at the end of the question paper. If it's not attached to where the question is, go back to the end of the question paper. That's where you're going to find it. It's always there. They always give, because you can't find the p-value on your own. So now we can continue. There are 5 questions attached to these calculations. So question 1 is saying choose the parental type. Choose the parental type. So the parental type, we know that there's nothing like thinking in this case, guys, or you getting this question wrong. No, there's no need of you thinking. Just come back here. What was the cross? The cross was between this parent. You are now focusing on what cross did you conduct to come up with the progeny. Forget about the F1. So the cross was between the F1 fly, which had this genotype, okay, and this one with this genotype. So now these were the parents. These were the parents which have given us these progeny. So now just interpret these in terms of phenotypes. This one, what is going to be the phenotype for this one? So you know that where there is a dominant B, this is going to be black. So the body is going to be black. Then there's dominant L there. It means it will be black with long wings. What about this one? They are all recessive. So these are going to be white with vestigial wings. So these are the parents. So that one should be the answer. One parent should be black with long wings, and the other one should be white with vestigial wing. That should be our answer because it's a dihybrid cross. So blackboard and vestigial wings. So this is not a dihybrid cross. This is a monohybrid. This is also a monohybrid. So the two can be answers here. So just check for the one which has got black body and long wings. So black body and long wings. The second one, black body and long wings. Yes, which is this one. And the other parent was white with white body and vestigial wing. So it's this parent. So the answer here has to be option 2. That's how you would have answered this question. It's very simple and straightforward. No need of you think, just interpolate the genotypes which you have. That helped you to come up with the progenies. Then question 2 is saying the calculated chi-square value is 50, 7500, 150, 200. So what was our chi-square value? Our chi-square value of calculated that is 200. It doesn't miss, guys. So the answer to this question is 200. So now you are finding it very easy to answer this question. So that's a 200. We have already calculated. Then 3 is saying the degrees of freedom for this test are. The degrees of freedom for this test are. So what did we say? Degree of freedom has got a formula. Degree of freedom is equals to n minus 1. So n was 4. 4 minus 1. Our degree of freedom is 3. So the answer has to be 3. So the answer is 3, not this one. No. N is just the total possible number of traits. So you can't say that is the degree of freedom because you subtract 1 from the possible number of traits. So we are getting a degree of freedom of 3. And earlier on I told you to say that most of the times dihybrid cross, the degree of freedom is going to be 3 because from dihybrid cross you are always going to have 4 phenotypes. And when you subtract 1 from 4, you always have a 3. whereas from monohybrid cross, the degree of freedom most of the time is going to be 1 because the phenotypes there are going to be 2. So 2 minus 1, you are going to get a 1. Then 4 is saying the tabulated or critical chi-squared value is. So this is the one I was telling you to say you can answer even before calculating the chi-square. Because this one, as long as you know how to use the critical table, the statistical table, which is, where is it? Which is this one here. You're just going to go at 5% and at the degree of freedom, which is 3. So we obtained that the p-value of 7.8. That was our answer. So the answer there is 7.8. That's the p-value that we obtained from this table because it's there. So that's the answer there. Do we have 7.8? Let's check. So do we have 7.8? Yes, it's option 2. So the answer is this one here, option 2. Then the last part to this question, which is question 5, reads, the results of the chi-squared test suggest that, so since we have seen that we had the chi-square of 200 and the p-value of 7.8, what does this suggest? So 200 is greater than 7.8. The results are going to be rejected. They do not fall within the expected Mendelian ratios, and there is a significant difference between the observed and expected values because chi-square is greater than the p-value. So these are some of the conclusions that we can make based on these values which we have. So the results of the chi-square test suggest that the difference between the observed and expected results is not significant. This is false. This is false because the chi-square is greater than the p-value. So when the chi-square is greater than the p-value, it means there's a significant difference between the observed and the expected values or results. 2 is saying the difference between the observed and expected results is significant. This is true. This is what I've been explaining or pushing out ever since I started this problem. Then 3 is saying the 2 genes are on different chromosomes. So this has to deal with linkage. We have not been talking about linkage here. So because the chi-square is greater than this, greater than the p-value, it means there was no gene linkage. There was no gene linkage, and if there is no gene linkage, it means that the genes are on the same chromosome. So this is a false statement. Then 2 is saying the 2 traits are controlled by multiple areas. So this is outside what we are discussing. We are not discussing about multiple areas here. This is not related to the problem we have in short. So multiple areas, a good example is when you're dealing with blood groups. So now that blood group is controlled by 3 areas. Blood is controlled by 3 areas. That is A, B, and O. So these are multiple, more than 2, both controlling a single gene, which is blood. So this is not related to our problem. The 2 characters are controlled by quantitative traits. So quantitative traits, these are also not related to the problem which we have. So the conclusion that we are going to make based on this result is option 2. There is a significant difference between the observed and the expected results. So that's how you handle problems relating to chi-square. So go ahead and find the chi-square problem and try to follow all these procedures that we have followed here. Then you're going to answer those questions.